Good morning. I'm Allison Shipp, Deputy Regional Director for Science and Research for the Rocky Mountain region. Welcome to the Spring 2025 Saline Lake Ecosystems Integrated Water Availability Assessment Seminar. We presented our activities from fiscal year 2024 sampling events in a series of seminars last fall. You can find those presentations on the Saline Lake Ecosystems website. Today's seminar will include a brief project overview, an update on 2025 sampling plans from each of the science pillars, and time for questions and answers. The Q&A portion of the seminar will not be recorded. However, this video will be available on the Saline Lake Ecosystems webpage after the seminar. Understanding if water management decisions can sustain or increase water bird populations in the face of hydroclimatic change and complex water use dynamics is critical to land, water, and resource managers. Determining the potential implications of increasing hydroclimatic and anthropogenic pressures on terminal lake ecosystems, water quality, and water quantity, and the resultant water bird habitat resiliency is essential to answering these questions and are the science pillars of the Saline Lake Ecosystems IWAA project. The project includes an outstanding team of scientists from many of our science centers located in and around the Great Basin. In 2022, this team brought together partners to help us develop a science strategy to understand how the terminal lakes within the Great Basin function as individual ecosystems and together as a system of ecosystems to support migratory water birds. One noted change in our team is the retirement in December of Mike Casaza, the lead scientist for the water bird movement pillar. Corey Overton is now the lead scientist, with Mike providing support as a USGS scientist emeritus. The team designed, with input from partners, a series of research questions that form the foundation of our science pillars. To address these questions, the project is collecting data and conducting analysis to better understand water availability and water bird habitat conditions and use. This information will provide land and resource managers and decision makers with a more informed understanding of terminal lakes and water bird resiliency. Today you will hear from each of the 5 science pillars: water bird movement, water bird aquatic prey food resources and habitat, water quality, water quantity, and remote sensing. Each pillar's lead scientists will present their plans for field activities beginning this spring and running through early fall. The overall data collection plan uses a categorization of the terminal lakes into intensive, intermediate, and limited categories. These categories were developed as part of the science strategy. For the intensive and intermediate lakes, the same types of data for each pillar will be collected, but at a different time step. All of the lakes will have remotely sensed data to measure the extent of the water. For those lakes in the limited category, the remotely sensed data, along with data from bird movement, may be the only data collected during the 5-year study. However, we are assessing the sampling design at the end of each field season to ensure that we are capturing data to address the priority questions within the existing budget. The team met in person in December 2024 to discuss the lessons learned, to inventory successes and challenges from 2024, and plan and coordinate the upcoming and future year field seasons. We also had a discussion with our Fish and Wildlife Service partners to further facilitate coordination between our two bureaus. We greatly appreciate their assistance with access to sites and their expertise on the landscape. One exercise we conducted at our in-person meeting was to gather around physical maps to coordinate potential instrumentation and sampling locations to create efficiencies for the project. In the next part of this seminar, each Science Pillar lead will present their plans for the upcoming 2025 field season. All plans are preliminary and subject to change as we await an appropriation for this fiscal year. We will hear first from the Waterbird Movement Pillar, followed by the other pillar presentations.
Hello, everyone. I'm Corey Overton, lead scientist for the bird movement task of the Saline Lakes Integrated Water Availability Assessment out of the Western Ecological Research Center's Dixon Field Station. Two of the key goals of the bird movement task are firstly to understand how the saline lakes operate as a network of habitats to support the ecological needs of migratory species across the annual cycle, and secondly, to develop an understanding of how the varying hydrologic, food resource, and remotely sensed habitat conditions that exist across this network affect bird occupancy and movements. Due to the variety of species that use the region, we limit the scope of our work to representative species that use different aspects of the system, such as species with different diets and behaviors, or that use the Saline Lakes region for different life history stages. Thus far, our work for the Saline Lakes project has focused on American avocet, which we have marked with GPS transmitters for the last 2 years, and Wilson's phalarope, which were marked with satellite transmitters and also tracked with MODIS radio beacons. Our plans for this year are to continue collecting location data from the already marked avocets and add those data to our location dataset, which contain over 1 million data points so far. We plan to expand our collaborations with other groups collecting movement data and to include data from other species, such as white-faced ibis, sandhill crane, various waterfowl, and pelicans. This year, we will also be marking 2 new species for the Saline Lakes effort, pelican and black-necked stilt. Our plan is to mark 40 individuals of each species at the intensely monitored areas of Great Salt Lake, the Lahontan Valley, including Carson Lake and Carson Sink in Stillwater, and various lakes in Central Oregon, including Summer Lake and Malheur.
Welcome, everyone. My name is Garth Herring, an avian ecologist with the U.S. Geological Survey Forest and Rangeland Ecosystems Science Center in Corvallis, Oregon. Today, I'm sharing our 2025 plans for the Terminal Lakes Waterbird Prey Availability Pillar. We are trying to understand prey availability within Great Basin terminal lakes with our research. The overall goal is to use estimates of prey energetics, habitat availability determined by the remote sensing pillar, and shorebird energetic needs to estimate lake shorebird carrying capacity, or how many birds a lake can support and for how long. Our estimates could then be used to evaluate how changes in lake volumes or extent and water quality would influence prey availability, with consequences for shorebird carrying capacity and potentially waterbird movements both within and across lakes. Note that prey availability is critical to most aspects of the annual waterbird cycle, including movements and habitat selection, reproduction, physiology, and survival. We largely focused on the lakes that were identified from the literature as important lakes for shorebird migration. The lakes include Malheur, Avert, and Summer in Oregon, Honey and Mono in California, Carson Sink, Stillwater, and Carson Lake in Nevada, and Great Salt Lake in Utah. These lakes also represented a salinity gradient from freshwater to hypersaline, with replicates within each salinity category. Our shorebird prey sampling methods use a repeated sampling framework where we collect invertebrates at specific lake depths tied to shorebird foraging depths along the shoreline. For shorebirds like phalaropes that are not tied to shallow waters, we are sampling open water sites. These open water sites also allow us to understand prey availability for important open water birds like eared grebes. Great Salt Lake, Lake Ebert, and Mono Lake support over 90% of the North American population at some point in their annual cycle. Finally, we are collecting water chemistry data at our sampling sites to understand invertebrate responses to changing water chemistry across space and time. During the 2025 field season, we will continue to conduct monthly repeated sampling at the 9 terminal lakes sampled during 2024. Additionally, we plan on refining some of our sampling methods to better estimate densities of certain invertebrate groups like brine fly larvae. We also plan on sampling at sites where shorebirds are actively foraging across a limited number of lakes to better understand their habitat selection processes. These active foraging sites will be determined from the movement data collected by the Bird Movement Pillar.
Hello, I'm Christine Rumsey from the Utah Water Science Center. I, along with Casey Smith from the Oregon Water Science Center, are the lead scientists for the Water Quality Pillar. Our pillar's focus is to assess the physical and chemical properties of the water, commonly referred to as water quality, in Great Basin terminal lake ecosystems, and to understand how these properties vary with changing hydrologic conditions. Water quality both informs our understanding of hydrologic processes and influences food web characteristics and prey availability for birds in a given habitat, acting as a key nexus between hydrology and ecology. Dissolved oxygen, salinity, temperature, and nutrients are all components of water quality. Water quality in the terminal lakes of the Great Basin ranges from freshwater, like Malheur Lake in Oregon, to hypersaline, such as Great Salt Lake in Utah, and from turbid, like Honey Lake in California, to clear, such as Walker Lake in Nevada. Water quality reflects stressors on the system. We also know the food web composition changes as a function of salinity. Water quality, and in particular salinity, provides the foundation for which organisms can survive in a given habitat. Water quality is dynamic in terminal lake ecosystems, and our overarching goal is to understand water quality properties throughout the Great Basin and through time by collecting both discrete or one-time and continuous water quality data. The key science questions driving our monitoring efforts include the following. First, what are the water quality conditions of Great Basin terminal lake ecosystems, and how do those conditions change across the region and during different hydrologic conditions across multiple water years? Our primary water quality topics of interest at this stage include salinity and water type, as well as the processes affecting water quality. Second, how does water management affect water quality conditions in impounded wetlands? Impounded wetlands are those created by some water control feature, such as a dike or berm. Third, what are reliable, easy-to-measure proxies for salinity? For example, can water density or specific conductance be used to reliably approximate salinity concentrations across the range of water types in the region? This will inform monitoring strategies moving forward. Fourth, how does water quality change as it moves through a system and through different seasons? This helps us understand how to collect representative water samples across the diverse habitats that occur in the region. And finally, what water quality conditions are ideal for phytoplankton populations that are the base of the food web and macroinvertebrates?
In 2025, we plan to deploy continuous water quality monitors that measure specific conductance, water temperature, dissolved oxygen, and pH. In addition, we'll collect continuous wind and depth data to pair with the water quality data. Water quality monitors will be deployed in terminal lakes, including Summer Lake, Lake Ebert, and Malheur Lake in Oregon, and in Carson Lake, and in the downstream terminus of the Stillwater National Wildlife Refuge in Nevada. The water bodies that will be continuously monitored in 2025 range in salinity, habitat characteristics, and water management practices. The continuous data collected this year will increase our understanding of how these systems change over time, and will link water quality, local wind, and water depth to the base of the food web. Lakes will be monitored from spring through fall.
2025, the discrete water quality team will repeat measurements of water chemistry and salinity across 15 terminal lake ecosystems in the Great Basin. Detailed water chemistry helps us better understand hydrologic processes, drivers of salinity change, and the concentrations of specific elements of interest, such as chloride or sodium, that may affect biota abundance and diversity. We focus on salinity because it affects aquatic habitat condition and the environments organisms live in. Water samples and in situ measurements will be collected in the spring, summer, and fall of 2025 across study areas in Southeast Oregon, Eastern California, Nevada, and Western Utah. In situ measurements of dissolved oxygen, pH, specific conductance, and water temperature will be collected across a wide range of site locations to characterize spatial variability in water quality and to better understand how it evolves from freshwater sources through a terminal lake or impounded wetland habitat. Water samples will be collected at a subset of these sites to advance understanding of geochemical and salt cycling processes. We will work in tandem with the water quantity and prey availability teams in areas such as at Lake Ebert, Summer Lake, Stillwater National Wildlife Refuge, and Carson Lake, where our pillar efforts overlap. Aside from data collection activities, we are planning to publicly publish a USGS data release of 2023 and 2024 water quality data.
This is Scott Heenick at the Utah Water Science Center. I will give you a brief overview of upcoming activities for the water quantity pillar. Our task is to determine how much water is needed to support bird habitat and their use by birds. As a reminder, the objectives of our task are to develop data sets needed to quantify water budget components, measure fluxes of water in areas identified by bird tracking, and estimate the residence time of inflow waters to determine hydrologic response times and inform hydroecologic resiliency. We are gathering basic data sets to support linkages between water flux and water bird ecology. Of primary importance is understanding the amount of water required to support ecosystems, as well as the resilience of these systems. There are three focal basins where efforts among all saline lake ecosystems science teams are coordinated. Work on the lower Bear River, the major inflow to Great Salt Lake, continues. Work on Lake Ebert and the Shiwakon River and Summer Lake in southeastern Oregon is ongoing. Water quantity work in the lower Lahontan Valley of Nevada, including Stillwater National Wildlife Refuge and Carson Sink, will commence this quarter. We plan to use the same monitoring techniques we used last year for this year's sampling season. At Bear River Bay in the Great Salt Lake, we will continue synoptic flow measurements and use field observations to identify sites for further research in fiscal year 26 on surface groundwater exchange and on water budgets of managed impoundments. At Lake Ebert, we will continue to make discrete flow measurements at the sites we established on the lake itself, as well as on the Shiwakon River, Crooked Creek, and Groundwater Springs sites. To gain a better understanding of consumptive use, surface water groundwater exchange, and hydroecologic resiliency, we will monitor inflows and collect water samples for isotopic tracers at all sites. At Carson Lake and Stillwater, we intend to monitor changes in inflows and salinity in areas of high bird usage. With newly acquired 3-depth LiDAR data, we will derive new high-resolution bathymetry.
Good day, everyone. My name is John W. Jones. I'm a member of the Hydrologic Remote Sensing Branch of the U.S. Geological Survey's Water Mission Area. It is my pleasure to share with you this year's fieldwork plans for the surface water and wetland remote sensing component of the Saline Lakes Integrated Water Availability Assessment. There are three overarching objectives for the remote sensing pillar. The first is the evaluation of accuracy and fitness for use of USGS and NASA operationally generated lake and wetland inundation extents. which not only provide current information on inundation extent, but also multi-decadal time series of inundation frequency. The second objective is to combine the extent data with water level and/or bathymetric data to estimate lake volume changes for use in water quantity studies. And the third objective is to extend what we learn through our intensive study sites and efforts to lakes throughout the ecosystem. Data collection takes two forms. Water quality and prey availability team members record the GPS locations of water's edge and inundated vegetation as they perform their fieldwork. Low-cost temperature and inundation sensors, as well as mobile network-connected field cameras, record inundation and weather conditions from spring through fall. The remote sensing team has been operating a camera at Summer Lake for 2 years. We are working with the Forest Service for permission to operate a camera from Fremont Point, which overlooks Summer Lake from the west. We are also seeking input from partners and others on whether there is interest in installing our temperature and inundation sensors and cameras at Carson, Stillwater, and Malheur. You can reach out to me directly at jwjones.usgs.gov or Saline Lake Ecosystems IWAA at usgs.gov for more information.
Hello, my name is Jessica Driscoll, and I am a program officer with the U.S. Geological Survey Rocky Mountain Region, and I am working with the science leads to coordinate the work across the science pillars to produce integrated science products. On the next several slides, I will review some of the products that the project has delivered so far. The project website can be found by searching for USGS Saline Lake Ecosystems. This website is the best place to find information about the project, including news, science, data, multimedia, and publications. A USGS circular was published in 2023. This aspirational science strategy describes a blue sky vision for a multi-stage and multi-agency process for integrated science implementation. The appropriated budget for the project did not allow for the full vision of this document to be realized. However, the importance of engagement with interested partners continues to be a project priority. The adaptive implementation framework described in this publication was co-developed with the Actionable and Strategic Integrated Science and Technology Initiative that continues to drive how we approach USGS science with partners. The full downloadable report can be found at the USGS Publications Warehouse and is linked to from our website. Fall of 2024, from October through November, the project provided updates and overviews for each science pillar. The recordings of these presentations by the science leads, as well as the transcripts, can be found online at our project website under News. In October 2024, a geonarrative named An Avocet's Travels was released. This interactive webpage follows the journey of an American avocet for 1 year, from July 2023 to July 2024, with the U.S. Geological Survey's bird tracking data. This page can be found under our project website under News. In December 2024, a journal article was accepted into the journal Bioscience, which was authored by project scientists. This publication describes and synthesizes gaps and describes research needs for waterbird conservation in the Great Basin. The analysis shows that integrated links between water availability, water quality, and food webs are lacking, something that the USGS aims to improve through the Saline Lake Ecosystems Integrated Water Availability Assessment Project. The article can be found online on our project website under Publications. The project has also produced several data releases, including a provisional data release. These data are accessible from our USGS science-based catalog and our project website. Additional water data are published through the National Water Information System and served through the USGS Water Data for the Nation platform. We will continue to approve and publish data following our USGS fundamental science practices and plan to link any datasets to our science-based catalog page and our USGS Saline Lake Ecosystem project webpage when they are fully approved. On behalf of all of our project presenters and all of the scientists working on our project, thank you for listening to our Spring 2025 virtual seminar and your interest in the USGS Saline Lake Ecosystems Integrated Water Availability Assessment Project.